Okay, and I'm start recording. Yep. Good afternoon, Dikshit, Deepit. I don't know. Probably not me. Okay, like we'll do one simple Manvita. Okay, we will do one thing. We'll discuss about the mentorship program once we end these things. Okay. Once we end the class, last time iruttalla aವಾಗ. Okay. Good afternoon. So it's already 3 o'clock. We can start the class. I think most of you. Yeah. I'm not running away. Good afternoon, Gopika. I told you guys to read about our second chapter and especially about the bacteria and its this one, right? Structure and all the things. How many of you guys completed? Click on yes or no. Completed yes, otherwise no. In YouTube also, I'm just want only 3 people are there. No issue. Just type it. Did you guys read anything? Okay, our notes. Okay. Not in a textbook. Textbook is also okay, but we made a detailed thing right there, right? You need to no tension. Once we complete this, then we are going for that. Okay. Why I skipped the unit 2 and came here, because these chapters are very important. If we complete these things, then it will be like less stress for us. Okay. So we'll start to revise these things and we can start doing the tests also, major test. Good afternoon, Anu. Okay, a few people, most of you are not at all responding. I don't know why. Okay, I'll leave it like that only. Good afternoon. I'm just starting our first question. We are not wasting any time. Okay, not a single minute also in this kind of classes. So before that, I'm muting everyone. Yeah, I muted everyone. Okay, so we'll start then. The first question came. This question will test you that how much do you guys read or not. Okay. The first question is, I think, easier one. The study of the cell is known as. They are asking, what do you call for the study of the cell? Okay. So I'm launching the poll. I don't think you need like time or anything, right? It's like a kind of answer you can give. No, you just answer in the polls. And who has came first time for the class? My name is Subramanya. I'm taking your biology classes. Okay. And whatever the question you have, just answer. Sorry, just put that in the chat box. I will read and I will answer to that. Good afternoon, Darshan. Really, guys, you guys are still giving wrong answer to this question. Huh? What we did in first chapter and second chapter. Okay. Finally, it's came to 60% some. Okay, I'm leaving today. Really. Okay. Every day I'm just watching 40% answer, 50% answer. I'm like, what? Hi, Venkat. Look, you should be happy, Venkat, because we started new chapter. Electricity came and it went. Okay. So it's fluctuating. Okay, I'm ending the poll in last 5 seconds. It is there. This site of a chapter is very important. It will this unit is very important. Okay, so the same thing will repeat some time in chemistry also in the biochemistry chapter, not biochemistry like polymer and biochemistry is there, right? There also it will come, and it will also help you to understand other things. Okay, so this unit is very important. Okay, guys, I'm ending the poll now, and I'm sharing the results. 80% chosen option A, 10% B, 6% C, and 4% D. Yes, and in YouTube, given A option. Okay, so exactly right. What is morphology then? Morphology means the study of the external character. Right, we discussed so many types regarding this. Right, metamorphosis, morphogen, all those things. What is karyogamy? Karyogamy means what? Can anybody answer? What is the meaning of karyogamy? Does anyone remember anything? Yes, exactly. Fusion of the nucleus. About fusion of nucleus. About nucleus only, but specifically fusion of the nucleus. We studied it in the how it will happen, especially in the fungi, right? There is like 3 different steps are there. So in that we studied this one. What is the meaning of anatomy? Anatomy means study of exactly internal structure. Okay, study of the internal structure. So it's the opposite. Like Anatomy deals with the internal things, and morphology deals with the external things. Okay. And what's the cytology? Cytology deals with the cell and its character and all the things. The correct answer is first one. Okay, it's easy one. Yes, study of cells, we can say. Exactly. You can also call this as cell biology, right? Both of the things are correct. Nothing is wrong. Cytology or cell biology, both of the things are correct. Okay. So second question, key. The prokaryotic cell are characterized by. They are asking how you characterize a prokaryotic cell. Okay, the options are absence of a chromatin material. Okay, absence of nuclear membrane, presence of distinct chromosome, and presence of distinct nuclear membrane. Yeah, some of you guys are already started giving answer, but some of you still need to think. Okay, don't randomly select anything, or don't think that by looking at some of the things. Hi Jagan, no issue, bro. So now only we started. I'm launching the poll. Exactly. I'm relaunching the poll, and who are in the Zoom answering the poll, and who are in the YouTube answering the YouTube live chat. I'm waiting. Yes, I got some answers. Yep. Shma, do one thing. Once go out of the class and join back quickly. Okay. We still will be discussing this only if screen is not visible. Exactly. Right. So you guys still have 30 seconds who not answered yet. So can answer now. Okay. Okay. Yes, I'm getting good answers. Only few people got confused. No issue. We'll solve that. And anyway, we are again studying these things one more time, right? So no issue that. I'm just checking. Have you guys read or remembered something or not? Haha. Good thing, Sushma. Okay. Yes, I'm ending the poll, guys. Okay. And I'm sharing the results. 2% chosen option A and 87% B. Nobody chosen option C and 11% D. It's a good progress. And in YouTube Live, everyone given answer B only. Okay, we will see which one is the correct answer. So first option, the prokaryotic cells are characterized by their things. So we have to try to find the character, which is actually the character or the leading character of the prokaryote. Absence of chromatin material, they are saying. Does chromatin material present in prokaryote or not? What do you guys think? Chromatin materials. Yes, okay. Some of you are saying yes. Okay. What do you think about chromosomes, guys? Is chromosome is present in prokaryotes or not? Chromosomes are present in prokaryote or not? Chromosome. Okay. Exactly. Okay. So what happens in prokaryotes? You think like this, right? There is no distinct nucleus. You think this as like a cell. What happens is like the DNA is like simply lying on the cytoplasm, right? So and another one thing is like it never form this kind of structure. It never forms a structure like this, what we can normally see as a chromosome, right? But it will never form this kind of structure, but it lacks a kind of protein called histone protein. We will discuss that later also. Okay. So we will use the term chromosome also, but we will also use the term chromatin also. Okay, the chromatin is present. We rarely use the term chromosome, but we can use it. No issue in that. Okay, because we are still in 11th and 12th, no problem in using those kind of words. Okay, so next, exactly, absence of chromatin material. It's not a correct answer, so you can remove this. Absence of nuclear membrane. Nuclear membrane will be absent. Yeah, absolutely correct. Absence of distinct chromosome. Okay, so it's like a partial. Yeah, exactly. Presence of distinct chromosome, you can't find it properly. Okay, we will keep this as like a partial one. Okay, presence of distinct nuclear membrane. No, there is no nuclear membrane, so you can remove this one. Okay, so you got two options which you are thinking which one is correct or not. Okay, so simple thing is characterization of the prokaryote happens by the absence of the absence of the nuclear membrane. Okay, so What is chromatin material? Chromatin material is simple. For now, think like this. We will discuss that later on also. For now, chromatin material, think that it is the condensed form or aggregated form of the DNA. Okay, that much is enough. So later we will discuss why we call it as chromatin, how the word came from, how we have to do experiments, what is U, hetero, all the things. Okay, so that's later on. Okay. So I think we can start the class. So some of you know the things already. Okay. So we'll go for some introduction. Okay. So from early on, we have a lot of confusion that how the living beings are distinct from the non-living beings. Okay. We have some answer now. Okay. Like we can say like there are some organism, it is non-responsive, like a plant. Okay, and there is some other things also there. This is also not responsive, like our house or the window or anything. Okay, so what's the similarities and the difference? That question arises, right? When you are saying a plant is a living, it should show some characters, right? To showing that character, it should have certain things, right? So what is that? We are trying to find that. Okay, so the ultimate answer is the cell. Okay, so cell is the fundamental structural and functional unit of the any organism, because whatever the organism may be, those organism is built by that particular chromatin, sorry, that particular, not chromatin. Okay, I'm just reading this one chart box. Built by the cell only. Okay, so you can say like whatever the buildings may be, it's built by a kind of mineral, right? It may be any mineral, that is secondary, right? There should be a mineral to build that particular building. Exactly, there should be a cell to build our body or any organism's body. Okay, but where can you differentiate a living cell and a non-living thing? Okay, because whatever the living things may be, whatever the cell may be, okay, maybe cell is a living. Yes, exactly, cell is living. Okay, like bricks of building. Exactly, Manvita. Okay, the cell is still made up of non-living things, right? So a non-living things, a group of non-living things join together in a certain orientation, in a certain order, and they form the living thing. Okay, so where you can drag a line. Okay, you can make a line in between like India and Sri Lanka, right? Even though it's there's like ocean is there, we will mark some territory like this much is Sri Lanka and this much is India, like that, right? But in here, this in between these living and non-living things, where you will differentiate, because non-living things only give rise to the ultimately give rise to the living thing, but we can't differentiate in between them. We still don't know. Okay, we still don't know where to put an exact line. Okay, so that's why what we will use, we will say that cell is a living. From there, everything came by. Okay, why we come to that thing? Because when you go for subcellular level, you will get confused. Okay, where to differentiate a living and where not from the non-living. Okay, so that's why we don't know. Still, we don't know. There are like a lot of hypotheses, a lot of things are going on, and we have like so many of books and all the things to differentiate between living and non-living. Okay, do you guys know like so many physicists to try to find these things? Okay, so but still like they are like in a like chaos. It's like a different kind of a chaos. Okay, so it will be like completely chaos, means inorderly form, right? Everything will be happen without any orders, right? You probably Vishal sir already talked about all these things, right? But living things is a chaos, but it's a ordered chaos. Okay, they will say like that. Okay, it's a ordered chaos. So completely, so many millions of trillion things will be happening, and everything will be happening in an order. That's the most amazing thing about the living things. Okay, but where it's initiated? It's initiated in the cells. Okay, it's initiated from the cells. Okay, that's what happens. So how, that's why we call it as a fundamental unit and a structural unit and a functional unit of the a living organism. Okay, what is this last organism? I didn't. Which one? Which last means, how can I say? There are like too many endings are here, right? So everything is, this one is the flying squirrel. Okay, this one is flying squirrel. This is a rat, I think. Okay, I don't know which is blue color it have. And this is, the squirrel. And this one is mouse and bacteria and fish. This one is mushrooms and all the things. So this one is a cell, final one. Okay. So these are the things. Okay, initiation. So that's the basic thing. Okay, we can say. So first, who actually saw the cell? When we ask this question, we will have like too many names. Okay, when gradually everyone is developing different kinds of the microscope. Okay, but lot of things are going on. That time, Robert Hooke developed the microscope exactly, and he started to watching different things. Okay, the easily available material for him is a cork cell. Okay, probably exactly. Probably you guys know what's the meaning of cork, right? So whatever cork is actually bark, skin bark cell. But if you ever saw a wine bottle, okay, if you ever saw a wine bottle or a bottle in your science class, okay, there is a rubber cork will be there. Okay, we normally call it as cork only, right? So that's a rubber one, but actually it's made up of the bark of the tree. Okay, yes. So it's easily available. He took a slice of that and exactly he slice of that and he observed in the microscope. Okay, don't ask me like who went there and took the photo. Okay, it's a photo taken later on. Okay, so Cork says discovered by the Hooke means this is what he looked under the microscope. Okay, and he called it as like a compartments, empty compartments. Okay, why he called it as a empty compartments? Because all of them are, what are these? These are all of them are sclerenchyma cells, and these are vacant or like empty sclerenchyma cells. Okay, that means dead cells, right? So that's why you call it as cellula. That means empty compartment. Probably you heard the term cell, right? In police cell, we normally call it. Why we call it as a police cell? Because it's a room, right? It's a room. It's an empty room where normally we will keep any prisoners, right? So in Latin it means empty compartment. Okay. That is later. That's later. Okay. So empty compartment, he called it. Okay. Later, the Leeuwenhoek actually observed the first living cell. Okay. Not only one, he actually observed so many cells. Okay. He observed protistomes. He observed some of the microorganisms and some of the living cells, RBC. Okay. He saw so many kinds of cells. So when we ask the question, who actually first observed the cell, it will be Robert Hooke. And if the question is like who first observed the living cell, then it will be Leeuwenhoek. Okay, exactly. Scrap of the teeth. Okay, so like he observed so many different living cells. Okay, now you guys tell me, what do you how are the neighbor cells are held together, sir? Yeah, we will discuss that, Shailaja. Okay, all these cells are joined by one together. Okay, exactly. There is a kind of membrane in between running between all the cells. Okay, so we will discuss the structure and everything later on. So I'm not want to fill your heads with a lot of new words. Okay, so now you guys answer me one thing. What do you guys think about the name of the cell? Okay. So is it correct or is it wrong? Is it something to do with or anything like any changes should be happened or anything? Cell, the name cell, what he given. Sometimes some of you guys think, right, probably if I got this name, it will be better, like all the things. Probably you guys thinking now only. Partially acceptable. Exact, sir, is this saw the cells with microscope? Yeah, they saw the cells through the microscope. Otherwise, they can't see, right? Exactly. They saw the cells through the microscope. Okay, exactly, Manvita, and all the other people also. So many people answered correctly. Okay, so the name word, the cell, actually came from the empty compartment. That means having a bound by certain things, a wall. Okay, but the actual thing is, neither it's empty, okay, nor it always binded by the wall, right? If you take any of your cell and check it under the microscope, it don't have any cell wall. right? And any living cell you take, none of that will be empty inside. They will contain protoplasm, they will contain the nucleus and all the things, right? So it's there, but the name continued from so many years and it's continuing only. It's like sometimes the people will have the names which is not at all suits them, right? Exactly like that. So it's a completely different thing. Okay. No, we can't change it because like it's so much well known now, right? That's why. Okay. Then after these things, so many people are working on. They want to give the cell theory. Okay. So there are like two people are important with regarding this. One is MJ Schleden and Theodore Swan. Okay. The thing is, both of them worked individually on botany and zoology, that means plant and animal cells. Okay, and they came together with a single statement. That means later they will work on with together, and they wrote this one, what is the paper, and they told that all living organisms, yes, all living organisms are composed of cells and the products of cells. Okay, they told whatever the happening is there, right? Whatever the living organisms are there, what you guys are telling, all of them are composed of cells only, and they are the products of these cells. Okay, these are the products of the cell only. They told. Okay, so this is called as first line for the cell theory. Okay, then what's the meaning of cell theory? The cell theory is simply like a theory to say what the cell and what's the its boundaries of the cell. Okay, what's the boundaries of the cell? Okay, so these are the first line. What they gave? They gave all living organisms are composed of cell, and what's the other thing? And they are the products of the cell only. Okay, they actually formulated it. Okay, so later one thing happened. This is not like complete or anything. Then Robert Virchow came and he told that omnis cellula is cellula. Okay, that means all cells arise from pre-existing cells. Okay, that means when we are discussing about Schledan and Swan, they gave the first line, but they don't know how the cells are formed. Okay, they actually thought that the in body the crystalline form of the inorganic substances will join. Substances will join and they will form the cell. They both of them thought. Okay, but Virchow told that no, it's not like that. It's actually all the words, all the cells what we are experiencing now. It's actually product of the older cells. Okay, remember, till none of them told how the first cell came by, right? None of them told how the first cell came by. They are still saying cells are made, organisms are made up of cells, and they are the product of the cells. Okay, and second, what they are saying, all cells are coming from the pre-existing cells. Okay, this much only given. This is what mentioned in the textbook also. Okay, so this is the first formulated cell theory, and these are the people who gave the first formulated the cell theory. Okay, but I don't think that cell theory is confined to this only. Okay, we gave so many other things also. Okay, so many other things means we added some other characters to the cell theory. Okay, we expanded the cell theory. Okay, all those things are happened. So all the organisms are normally confined to this only. You can ask, is there any exceptions? Is there? Yes, they do have the exceptions. Okay, what are those exceptions? You guys can think, right? Which one do you guys think is an exception for the cell theory? All organisms are made up of cell, and they will be the product of cell, and all cells are arising from the pre-existing cell. They are saying exactly the virus, the viroids, the prions. Yes, exactly. All those things are actually not belongs under this cell theory. Okay, sometimes what happens, there are like some other deviations also come. When we added the new things, we added something like all living cell will have a nucleus. Okay, nucleus means they will have some genetic material, and we also told that they will do these kind of functions. They will have plasma membrane, they will do this function and all the things. Okay, but there are some exception in that also. Okay, okay, Priyank, I'll get back to you. Okay, so what is that then? So if you take the RBC, Okay, if you take your RBC, then how can you say that? Because what do you guys think, sir? Do you think that, exactly, right? When you take your body RBC, this RBC don't have any nucleus. Okay, I don't know how many of you know or not. Your RBC don't have any nucleus. Why do you guys think that? Why do you guys think that the RBC don't have any, sorry, RBC don't have any nucleus? What may be the reason? Anyone? I'm waiting in YouTube also. To fill the hemoglobin, because it's not, hey, it's a cell only. RBC is cell only. We only told, right? RBC, red blood corpuscles, that means red blood cells. It's a cell to accommodate more oxygen. Exactly. Yeah, exactly. So their function is to transport the oxygen, nothing else. Okay, their function is to transport the oxygen, nothing else. So what they will lack, or they will lose the this one nucleus later on. Okay. What about the first cell? Exactly, Ashwin. Nobody told about it. Okay. Exactly. Okay. Not more hemoglobin. It's like doing their work. To do the work, they don't have any other work. Okay. And the second thing, so someone else, I forgot the name. Okay. So what is the use of, or what is the contribution of Malpighian cells with related to the giving cell theory? Okay. Malpighian cells, you can say, is our excretory cells. Okay. These cells helped Virtue to understand the division of the cells. Okay, division of the cells and how it acts and all the things. So by working on that, he actually gave this words, okay, gave this particular sentence. And as Ashwin asking, the first cells, we exactly don't know that how this complex kind of thing happened, but we still have some answers. Okay, so the answer is like, who, what, who is this? Prakriti, did you copy it somewhere or what, or you just typed all those things? Okay, correct. Okay, so so what happened in the initial days, due to the different kind of nature what we have? Okay, so a small lipid and small amino acids all are developed. Later they will become complex in a million of years. They form a cell, we will say, but we still don't know how a smaller, that means how can you say, a basic kind of cell, it's actually the bacterial cell. Okay, but bacterial cell is so complex to understand, we can't replicate it. Okay, you can't replicate a bacterial cell. Even though you replicate it, you can't make it self-sustain, right? So still we don't know. Okay, we don't know. We know that how it started, but we don't know how it went through a particular path to reach here. Okay, so it have a DNA or not? Which one, Chirant? I didn't understand. Which one you asking, having DNA or not? Okay. Can you form an artificial cell by adding no, that's what I'm telling you, we don't know. Okay, RBC don't have any this one DNA, it don't have any nucleus, means it don't have any DNA, right? So even RNA is also very few. Mature RBC don't have the DNA nucleus. Okay, the younger RBC will have, mature only don't have. Okay, so we will move on. We have a question, I think. Okay, the question K, yes, simple. Cell theory states that all cells are living, all cells possess nuclei, cells reproduce by mitosis and meiosis, cells are fundamental structural unit of plants and animals. Okay, I'm launching the poll because I don't think anybody need any time. So, okay, I'm launching it. So you can answer quickly, right? So it's also one of the easy question. But think, guys, okay, don't choose the option like, okay, it looked like the correct answer, so I will choose. Never do like that. Okay, I have to read all the options before choosing any one particular one. Okay, I'm getting so many correct answers. So many wrong answers also. We'll see. Got it. Amit. Okay, we still have 25 seconds. We will wait. Finally, like probably after registration, now the this one attendance is coming back to the normal. That's a really good thing. Probably everyone is completed UT and enjoyed 1 or 2 days and back to the classes. Ha, Venkat. Lifespan of a single cell depends, Nayana. Okay, it's always depends. So one of the cells may live up to the several weeks or months. Some of them will die in few minutes also. How the check cells are easy? You can remove it by cracking it. That will be there, Nandish. I will inform you. Okay. Yeah, I'm sharing the results. 14% A, 4% B, 5% C, and 77% D. No, for 11th, the final exam will not start yet. Don't take tension. We still have like two months. Okay. And in UTB, everyone gave the photons. Exactly. Cell theory, don't say that all cells are living. Okay. And it don't state like all cells possess nuclei, what we are dealing, that particular cell theory. And cell reproduced by mitosis and meiosis, they not even discovered mitosis and meiosis that time, okay? But they know that cells are fundamental structural unit of the plants and animals because Sladen and Squand gave the these statements, right? That's why. So the correct answer is option D. Okay, so next one, an overview. Okay, so what more we will discuss, the length. Actually, we discussed the same kind of things in the first class, right? There we discuss like how much small a living system from to be how much large it is, right? A smallest cell, we are remember, we are not at all taking any virus or viroids here. Okay, no, Insura. That means when they are giving Sladen and Scavan, they discovered about those two things, right? So that time cell theory is like that, and that old theory only what you guys are reading. Okay, new theory is different now. That is what that's not included in the syllabus. Okay, new theory have like a lot of fundamentals. Okay, in these things we are not at all reading about virus and viroids because still we won't consider virus and virus as a cell, right? So no, we don't see it as unicellular. We not at all see it as a cellular. Okay, we call them as acellular. Okay, we call the virus viroids as the acellular. Okay, so that's why we never mentioned any virus or viroids here. Okay, so smallest cell we will consider about mycoplasma only. So for the formation of a cell is the atmosphere, major factor. That one became a major issue, Jivan, actually. Okay, the environmental factor, how the environment is daytime. Okay, it's an oxidative environment. We will see it. Okay, we will learn that in the evolution later on. No issue. The temperature, the pressure, everything actually initiates the cell process. It can't start now. It won't start in this kind of environment. Okay, it's not oxidative environment. Okay. So these are smallest cell, that means mycoplasmas. So 0.3 micrometer. Okay. So largest isolated cell is egg of ostrich. Okay. So as you can see, mycoplasma belongs to the bacteria. Exactly. Mycoplasma belongs to the kingdom Monera, right? And the longest cell will be your nerve cells. Nerve cells can reach up to the several centimeters. Okay. That's why. Is it egg a cell? Exactly. Egg is a single cell. Okay. So egg is a single cell, yeah. But don't try to find. Don't think that the yellow color is the nucleus. Okay, don't think like that. Yellow is not the nucleus. All of them is food materials only. Okay, everything is a food material. Okay. So and size of the bacteria is 3 to 5 micrometers, and human RBC will be 7 micrometer in diameter. Okay, when we are normally discussing, we normally mention about these nerve cells because nerve cells will be longer in length. Okay, but you can consider the muscle cells. So muscle cells also comparatively longer, and they will be thicker also. Okay, those are the two major things, two major cells in our body which will be comparatively larger than the other things. Okay, and the smaller things you can think about this mycoplasma. Remember, do you guys know the other name for the mycoplasma? Do you guys remember anything? What is the criteria to define a cell? So we have some. Okay, we have some, the basic thing is cell theory. Okay, the new formulated cell theory is the basics. Okay, a cell, if you call something as a cell, it should have a membrane, okay, a plasma membrane, and it should be filled with something. That means either protoplasm, right, or something. And it should have some genetic material which can transfer from one to another. Okay, and it should have some organelles, like any organelles. In some of them may have, some of them not have complete one. So these are the some of the criteria. What we see, the characters, right? Character of the cells, only criteria for the cells. Exactly. We have the name. One is the PPLO, pleuropneumonia like organisms, or you can also call it as mycoplasma like organisms, right? MLO also you can call it as. These are the smallest cell, right? So just imagine from smallest cell to the several centimeter long or several millimeter long, how the different length of cells are there. Okay, you guys can ask, sir, why cells can grow so much big? They can have the potential and everything. Why they are not giving? Why they are not growing big? Okay, the issue is like how they maintain themselves. Okay, I will give you an example. So probably I gave this example already, right? Probably in the initial class I mentioned this. Why do you guys think that our India is divided into several states, several districts, several taluks, several gram panchayatis, okay, several wards? What is the necessary of that? They can combine everything and they can rule from a single place, right? Without having any changes or without having any compartmentalization. Exactly. To make administration easy. Exactly. Large country means a problem will happen, right? Large country, think that this is okay, think that something, okay, something our country only, okay, this is a rough diagram, don't offend, okay. So it's completely, so think that our Delhi is here, okay. So what happens, there is no communication or anything, as one should be there. Think that some riot is happening in this part, okay, in Karnataka. So don't call it super, okay, I know it's worse. Then the police first come from here to here. How much time it will take? Okay, even though to arrange a flight, if there is no flight, it's like completely what they have to take a train, and train will take minimum 1 or 2 days. Okay, so it's like so much thing, right? They can't do it. Exactly same thing happens. The more there are a lot of things will be there. Pawan, 1 second. Okay, so I will answer to that. So a lot of things will be there. The issue is like the most active cells are the smaller cells, okay, because this nucleus can distribute, or this nucleus can do the things or make the things easily, because the periphery is solo, okay. The most not active cells are larger cells, okay. If you take an example of the egg is completely filled with all nutritive materials, okay. It's not completely active one. Okay, but if you take an example of zygote, the developing zygote inside to that, okay, later on, if it's fertilized, then it will be more active and it will be more minute also. Okay, if you take an example of any stem cells, okay, stem cells, it may be meristematic cells of the plant or the animals. Those cells are very small because they are keep on dividing, okay, because to maintain that, okay. So, what is the size of the egg of the ostrich? Just imagine here only. So same length, our almost within our hand. Okay. That is virus don't have anything. That's why it will do that. Okay. So Lipika, I answered this question. So the major things on our body, in my opinion, like we will normally call it as a nerve cell, apart from probably the muscle cells. Okay. So and another one thing, why some of the cells will have two nucleus? No, amoeba normally has single nucleus. Okay, amoeba normally has single nucleus, and there are like a lot of strains are there, lot of cells are there. It may have more than one nucleus also. It's normally to redistribute their function. Okay, they can't divide, or they can't do multicellular structures, so they will go for some other ways. Exactly like dikaryon stage, what we saw, right? Exactly. And also in like we also see xenocytic kind of states also, right? So what happened, instead of they divide the cell, they made several nucleus copies, so they can like reach out to things, right? Think that this is the large cell and it have only nucleus, but it can't reach up to here, so it will divide and it will form another nucleus, place in it will place four nucleus in four corners, so it can, everything is settled now, right? Everyone can take care of their compartment properly. So that's also they will do. Okay, they will do different kinds of things to maintain that things. Okay, to maintain the structure and all the things. Okay, and exactly. So we will move on. Okay, so how will be the function, shape, and everything? It's always vary. This is not like universal thing. Like it should be like this. It should follow these kind of set of rules only. Okay, there is nothing like set of rules in biology. Okay, so that's why I told in what happens in physics when something is like not going according to the rules, right? So then what happens, it will lead to the some other, like normally in physics everything follow the rules. It's very rare you find something which you do not follow the rule, but if you work on properly, then you will get how it's happening, right? It should be follows one or another rule. But in biology nothing will happen like that. Okay, not like exceptions, it's like completely different things. Okay, the large cells like it. Yes, actually. And more functional cells also have more multi-nuclear stage. Okay. So in biology, what happens? So these cells, every cells will tend to have differently or work differently. Okay. As you can see, if you see the structure of the cells, some of them will be circular, some of them will be longitudinal, long. Okay. Some of them may be having different kinds of shape. You can compare with a nerve cell, as you can see here, to the muscle cell, and to the so many bacteria, and to the sperm, and to the egg, right? So many changes are there. The sperm will have the flagella, the egg won't, right? In human, I'm telling. Okay, the plant cell will having a kind of a shape, but animal cell don't, right? And in our a single body only, you can find so many kinds of cells. Muscle cells only, you can find three types of the muscles, right? Blood cells only, you can find two major type of the bloods, RBC and WBC. In WBC only you can find five types again. Okay, in WBC only you can find five types of the cells. Okay, so many differentiation are there depending upon their functionality and their environment. Okay, so no, Ashwin, mycoplasma and PPO both are same only. There is no differentiation. Okay, so many changes are there. Okay, so many changes are there. So mycoplasma, we call it like because it's having a different kinds of the shape and all the things. PPLO called it like it will cause pneumonia like diseases. That's why you call it as like pneumonia like organism. Both of them, even ML was also same only. Okay, mycoplasma like organism. Okay, so yeah, so many different types of cells will be present. Okay, but when you go for like a broader level, we can differentiate into two types. One is the prokaryotic cell and another one is the eukaryotic cell. What is prokaryotic cell? Prokaryotic cells are older cells with a simple, comparatively simple kind of the mechanism. Okay, eukaryotic cells are the new cells with more complexity. Yes, more complexity. Exactly. You can call it as the primitive and new also. Exactly. Okay, you can call it as primitive. So understand, right? The prokaryotics are the first organized cells or primitive kind of the cells, but complexity is there. But when compared to the eukaryotes, they'll be like less complex. Okay. Yeah, there are like so other things also there. No, Mohan. Yesterday only we discussed. Where are you that time? So once fertilization will happen inside the body of the this one bird, later the shell will form. Okay, then the egg will comes out. So it's a step. First, our regular sperm will be regular egg will be there. What we discuss in humans and everywhere, right? Exactly similar kind. The sperm will enter and it will reach the egg and it will fertilize it. Then the body of the female will cover that particular zygote with all the nutritive materials and it will cover with a shell and it will come out of the body like that process. Okay. So there are like some other differences also there. That means how a membrane bound organelle don't have the membrane bound organelle. So many differences are there. We will learn that in the later one. Okay. So one question came. We'll solve this question. Okay. So this actually tells you that how much you guys read or not. Okay. So pneumonia like organisms were discovered by. The options are Emil von Behring, Knockout and Rocks, Martinez Benzernik, Colton Gestusik. Okay, so don't choose it randomly. Okay, when you choose randomly, I will got to know. Okay, so you guys are like throwing a stone. We will say, right. It may hit the mango or it may not. If it hits, you will say that I perfectly make a this one what is called point and I throwed it all those things. If it won't hit, I will say, oh, probably it only the wind probably blown. That's why it actually tilted all those things. So don't do like that. Okay. I'm just launching the poll. Really? Think one more time. Exactly, Mohan. Really, guys. How is this even happened? I'm waiting in YouTube. At least there I will get the more familiar answers. I'm getting the correct answer in the chat box, but I'm not getting the correct answers in the poll. Okay, we'll see. You guys still have 30 seconds. We'll wait for. Okay. I came to know, bro. I know. So many of you haven't even touched the notes. Okay. If you touched the notes or if you revised it, you could have answered this question correctly. Okay. Without any problem or without any hesitation. Fluoropneumonia organism, where TMV came. TMV is a virus, tobacco mosaic virus. Okay, I'm ending the poll then. Nobody's answering. Only 55. Really? I'm sharing the results. 10% chosen option A, 22% chosen option B, 65 chosen option C, and 4% chosen option D. And nobody actually given any answer in the YouTube live. I'm just waiting. Okay. And don't think that it is not like democracy. Okay, we are not running the democracy. So what happens? Not highest poll will not always not always will be the correct. Okay, I'm not discussing anything here. Okay, I'm just giving the correct answers, and you know guys know that the correct answer is Knockout and Rocks. I don't know who is that Martinez Benchernik. Huh. So yeah, just like you guys made me confused. Okay, so what's happening? All the things. MW Benzernik. Okay. So probably you may have you guys only think about it. Okay. So what is the homework for? Don't think that class is closed. Okay. So don't think that class is closed. Your homework is read Kingdom Monera, its characters properly. Don't do it like today. Okay, so read it properly and come to the class. So then we will run the class properly. If you guys give this kind of like direct and easy question or wrong answer, then it will like our heart will ache. Okay. Mohan, I told you it should have a membrane, not cell wall. Even our body cells also don't have a cell wall, right? Still it's a cell, right? But it's have a cell membrane, exactly. Same time, the mycoplasma will have the cell membrane, but not the cell wall. So you can call it as a cell. What Venkat. Okay. So we'll move on to the prokaryotic cell. Once we complete those things, readings, and we will discuss about some other things also. Okay. The first thing about the bacterial cells. Okay. What about the bacterial cells? The bacteria or the bacteria means, or when you think about prokaryotes, we will normally discuss what the major three things. What are those include? They include bacteria. What this bacteria include then? This bacteria will include either a archaea or archaebacteria, or it may be a eubacteria. And even though it is included in the eubacteria, it shows some difference, that is cyanobacteria. Right, cyanobacteria is included in eubacteria only, but I'm writing it here because like it's a little bit different, right? So the changes are there. These are the actually soul members which actually having a prokaryotic kind of the configuration, right? So a big kingdom. When you compare this one five kingdom classification, right? So in that the shape will be vary always, and they have highly diverse and they are present everywhere, and they are very small, right? They are very tiny and they are very rapid. I told you, right? Our tiny will be more rapid. Okay, exactly like this. So all of these will be tiny and they will be more rapid. So example, you can take any kind of bacteria or cyanobacteria or mycoplasma or PPLOs. Okay, here you can observe the cyanobacteria may form or attach one together and form a colony-like structures. Okay, colony-like structures. Don't think that these are multicellular structures by looking at them. Okay, they are acting individually only, but they are like joined together. I told you, right? Colony is like your colonies. Okay, we have colony like that colony, Saint Teresa colony, Gandhi colony, something, something, right? In that you can find hundreds of houses, and all of them are not relatives, right? All of them are not sharing the food and all the things always. But what makes them living in particular things? Their houses are attached, right? Their houses are attached in a single road, connected by a single road. That's why you call it as the colony, right? Exactly. Colony means that much only. It's not like they are working like a multicellular. They never work like a multicellular. They are unicellular individual cells, but they live one beside another one thing. Okay, like apartment also you can say. Okay, rapid means what? Rapid means they are so speed. Okay, like you can call it, rapid means speed, quick. Exact. You only know the answer. You only ask question. Okay, rapid means quick. Rapid means in speed or quick. So they will do the reproduction in a quick manner. Okay, always they will divide and give rise to new cell or new organism, and they will be like keep on dividing, keep on growing. That will be happening. Okay, rapid fire game. Okay, that's what happens. So next, when you go for the shape, and you know that we have four major shapes. Okay, one is a bacillus, another is a coccus, vibrio, and spirillum. Bacillus means these are rod shaped. Okay, a kind of a rod shape will be there. And second one is coccus. Coccus means it's like a grapes. Okay, circular or spherical shape will be there. Vibria will be a comma kind of shape, as you can see. Okay, where is the comma? Okay, something like this. And spirillum will be spiral shaped. Okay, we discussed about all these things. And you can, vibrio and spirillum normally present individually, but if you take bacillus and coccus, you can find their aggregated forms also. If single is present, you will call it as monobacillus or monococcus. Two is there, diplococcus and diplobacillus. Three is there, triplococcus and triplobacillus. If it is formed in chain, you will call it as streptococcus and streptobacillus. If it is formed in a group, so you call it as staphylococcus and palisade bacillus, right? So all those things will be there. Okay, vibrio are flesh-eating bacteria. No, normally vibrios are disease-causing bacteria. Okay, all these different kinds of bacteria, it's not like flesh-eating. All of them are like depending upon feeding on the dead and decay materials normally. Okay, when they are become pathogens, so they normally eat something which is present in there, right? It may be if it's an infection, they're actually feeding on your skin only. So that's in that manner we can say. But when we say flesh eating, we normally say it for like which can directly eat flesh, a higher organism. Okay. This is the only thing left in my life to hear. Okay, corona is coccus. Corona is a virus, and you named it as like coccus. Okay, so yeah, I know you made a joke. Okay, I simply told. So differentiate. Okay, don't intermix the Monera, Protista, virus, and all the things. Okay, so these are restricted to the bacteria only. Okay, so that much. We will go for the next one. The cell organelles, what are these present? These have very minimalistic kind of the cell organelles, right? So what are the things present in here then? The major things present here is plasma membrane. So it is present. So, and it may have cell wall, right? It will have a cell wall and also glycocalyx layer. We will see that. Okay. So you can call it as membrane also. Okay. What it will have apart from this, it will have a ribosome and it have the nucleoid, or you can call it as the nucleoid, or you can call it as the genetic materials, right? And what are the other things it will have? Any other things we missed it? Exactly, yeah, it may have some extracellular ribosome. Yes, it do have the ribosomes. Yes, so it do have certain extra protuberances like flagella or fimbriae. I think you guys know about the difference between flagella, fimbriae, and pili, right? You know that, right? No, you don't have any mitochondria. Yeah, we will learn about all those things. Okay, one more time. That's why I told you guys to read about the things. So we will discuss one more time, but must read second chapter. Okay, otherwise we'll stuck here only forever. Okay, that's why. They do have the plasmid, and you guys know about the definition of plasmid also. Three-line plasmid. Sorry, three-line definition, right? So we studied everything, almost everything about bacteria in the second chapter only, right? So these are the major components. So you can also include cytoplasm, right? You can't miss the cytoplasm. It is not a cell component, but organelle. It's not a cell organelle. You can call them as like cell components. I will remove these things. That will be better. Cell component. Exactly. And there will be another one, changes or different thing will be there. That is what you call it as the exactly Mohan. Mesosome. We actually studied about this also, and it may have vacuole also. Okay, and it may also have inclusion bodies. We will learn about all this in detail, so that's why I'm not giving any details for anything. Okay, so we will see that in membranes. Okay, I'll just mention it like membranes, not plasma membrane. Yeah. No Golgi, no mitochondria, no chloroplast, because all of them are present in eukaryotes only, not in the prokaryotes. Okay, exactly, Lipika. Okay, so how can you define a prokaryotic cell then? Prokaryotic cell, a simple kind of cell which do not have any membrane bound cell organelles. Okay, what are membrane bound cell organelles then? What are you saying, right? What are you guys are normally will say? The mitochondria, Golgi bodies, or apart from chloroplast, okay, endoplasmic reticulum, okay, nucleolus and nuclear membrane, right? All of them are it may be membrane bound. Some of them are membrane bound, but those organelles, what you're normally familiar with, will not be present in prokaryotic forms. Okay, so it's not present in the prokaryotic form. I will come to that, Pawan. No issue. Okay, we will learn all those things. No problem. Okay, how it will produce the energy then? It don't have mitochondria. How it will produce food if we don't have the chloroplast? Okay, how it will cope up with that? All those things we will learn this in chapter. Okay, so first one, cell envelope. I don't think anything is left to explain in this one. So why you may ask, okay, why you are telling same thing to everything? Because we learned in detail about all these things in the second chapter. That's why I told you guys to read about things, right? So cell envelope is actually 3-layered. Okay, it's a 3-layered. So I will write it one more time. So these 3-layered can be arranged based on their availability in how much. So the outer one layer is the glycocalyx layer. Okay, outer one will be the glycocalyx layer, as you can see here outside, and next one will be the cell wall, and third one will be the plasma membrane. Okay, so how it will be different and how it will be useful? The first thing, the glycocalyx layer can be present in two forms. So either it may be a thick form, or it may be thin, right? So what you guys will tell when it's the glycocalyx layer is very thick? How you will pronounce it, or what you call it for that one? Can anybody answer? Exactly. we call it a capsule. We call it as a capsule. Glycocalyx, when it's thick layer, then we call it as the capsule. What you guys will call it when it's like thin? The layer is thin. Exactly. We call it as the slimy layer. Okay, we call it as the slimy layer. So what is the function of the glycocalyx then? What is the function of the glycocalyx? Anybody can answer? I'm just waiting. You just type up words, okay? I don't need long sentences, just to type the words. Exactly. First one, avoid desiccation, okay? So it will stop desiccating under the sunlight. Exactly. It will help to attach to the substratum, and it will help in the protection or mechanical and chemical damage to the cell, okay? So these are the major things which actually help them, okay? From what it is actually protecting the cells, it is protecting from the sunlight, it is protecting from the chemical environment. It may be the tablet you ate, okay, or it may be some acid in your stomach, okay, or it may be a strong alkali which is present in the nature, okay, whatever the thing may be, right? Exactly. Structural support, it will give the structural support, exactly. And when you go for the cell wall is made up of what? In bacteria, cell wall is made up of what? You know the answer, right? Second layer is the cell wall. When you are going from outside to inside, exactly, it is made up of the peptidoglycan, right? Peptidoglycan, not chitin, peptidoglycan. You can call this with another name. Do you guys remember another name? Exactly, murin, right? Murin. Okay, so Ananya, exactly. I will come to that. Now I will explain this. Okay, you answered the same thing when we are discussing about second chapter also, right? I remember. Yeah. So cell wall is made up of the peptidoglycan. This peptidoglycan is composed of what? This composed of two different contains. Okay, one is the NAG, another one is the NAM. Okay, what is the meaning of them? NAG means N-acetyl glucosamine, and what's the meaning of NAM? NAM means N-acetyl muramic acid, exactly. Okay, so these are the two components which actually compose together or mix together, and they will form a layer called as peptidoglycan layer. Okay, this peptidoglycan layer is very important, or this is the cell wall composition. Okay, if you go for the plant cell, if I ask you what plant cell wall is made up of, you can answer cellulose, right? If I ask the same question which related to the fungi, you guys can answer it's a chitin, right? So exactly, when you come back to the bacteria, and if I ask you what bacterial cell wall is made up of, then you have to answer it's a peptidoglycan, or you can call this as both the name are same only, murin. Okay, so what is the composition of the peptidoglycan or murin then? The composition of peptidoglycan and murin is N-acetyl glucosamine and N-acetyl muramic acid. Okay, both of them. Yeah, you can simply call it as Nag and Nam. Okay, you remember I like something Naga, Nama, Hakada, something. Okay, so Nag and Nam. So these are the things which are mixed together and they form the cell wall. Okay, and then we'll the plasma membrane. When you come to the plasma membrane, it's like simple but complex, exactly. Okay, so these are semi-permeable, or you can say that selectively permeable. That means these are only allow the things which are actually should go inside. Okay, not all the things. Okay, this is made up of the double membrane, right? It is also made up of the double membrane. So from what it is made up of then? It is made up of the different kinds of the lipids, or you can say it's like phospholipids. Act as a security guard. It will only allow the things which actually should go inside. Okay, not everything. Water will come and say that I will go inside. It will say, okay, you can't. Okay, and same thing, there is like lipid will come and it will say, okay, you are a lipid, right? So I will let you inside, like that. Okay, so it will make a discrimination. Okay, it will make a discrimination and it will decide what to go inside and what not to. Okay, but from inside only somebody will order, okay, I need water. So then what it will do? It will make a door for the water to come in. Okay, and it will send some people out. and those people will attach it to the water and they will bring water inside and they will leave it. Okay, that's what we call it as sporins. Okay, we will learn that in the plant physiology. That means it's a kind of gates will be there. Okay, so those are actually controlling the movement of the molecules. Okay, that's also happens. So those kind of things will be present and those are actually maintaining that. So whenever someone will ask you, we'll come back to that respiration, synthesis of lipids, we'll come back to all those things. Okay, so when anybody asks you about cell envelope, you have the answer like it's made up of three kind of envelopes. In outside there will be glycocalyx layer. Glycocalyx layer will be either thick or thin. If it is thick, it's called capsule, and if it is thin, it's called a slimy layer, and it's also a kind of lipid layer only. Okay, and second layer is our cell layer or cell wall, which is actually made up of peptidoglycan or murin. And the third layer will be plasma membrane. It will be double membrane system made up of a kind of lipids, and these are selectively permeable. Okay, these are the major things. So next, we'll move on to the next one. So based on the bacteria types of the cell envelopes, we actually divide into two types. One is a gram positive and another is the gram positive, gram negative. I think you remember the this one experiment, right? What he will do, Christian Gram, he took a stain called gram stain. Okay, what is the gram stain? Do you guys remember, anyone, the name of the gram stain? Yeah, Lipika. Name of the gram stain. Name of the gram stain. Which violet? Exactly, crystal violet. Okay, crystal violet. Exactly, crystal violet. So what he did, I will summarize the experiment in here. Okay, I will summarize the experiment in here. What he did, he took some bacteria. Okay, and then he put them in the crystal violet. Crystal violet, you can call it as the gram stain. We call this as the gram stain. Okay, then he added, for this bacteria, he added some iodine solution. Yes, he added 0.1% iodine solution. Don't think about it, think that iodine solution. Okay, then he washed with water, and then he again washed with the two things, either alcohol or with normally he washed with the alcohol. We will write down like alcohol only. Then he made an observation. Okay, he made an observation. Some of the bacteria, so some retain violet color. Okay, so what happened here, he took some of the bacteria and he put the crystal violet color. So all the bacteria got the crystal violet color. Then he put some iodine solution for better absorbing of that, adjusting or attaching that this one stain to the bacteria. Okay, then he washed that and then he put the alcohol. Okay, so don't think that while washing all the bacteria went away. We actually fixed the bacteria to the this one slide. I told you how we do that, all those things. Okay, so after treating with alcohol or washing with alcohol also, okay, here is also wash, not illa. What happened is like I took this particular I am not prepared the slides. These slides are prepared by someone else, right? They used this particular background. In that background, the font size is larger. Okay, I am irritating. I'm only getting irritated by the font size. Okay, so it's not at all looking good. So after washing alcohol also, so he observed and some retain the violet color. Okay, he called them gram positive bacteria. Okay, he called them gram positive bacteria. How this happened then? Okay, what is the reason for this one? As you can observe here, one second, guys. One second, stop it. Yeah. If you observe here, a Staphylococcus aureus is a gram positive. Okay, and Escherichia coli is gram negative. It do not retain that color. Okay, so what happened in here then? Okay, so these gram negative bacteria, will have the outer lipid layer. When you wash it with the alcohol, lipids are washed away. Lipids are washed away along with what? These are washed away along with the stain. So these do not have any color, but gram-positive bacteria do not have that much of lipid outside. So, and they have a different cell wall composition. So that's why what happened, they retained the gram stain. Okay. So what's the use of this? Using this, you can differentiate bacteria into two major groups. One is a gram-positive and another gram-negative. Then what's the problem with, how can you conclude anything with it? Yes, we can conclude something. What is this is, the bacteria, even though look same, may have the different composition of the envelopes. Okay, it may have higher lipid or lower lipid contents. And the second one thing, the gram negatives are normally pathogenic in function, more pathogenic, okay, compared to the gram positive. Okay, I will write down here only. This is gram positive. These gram negatives are normally pathogenic. Okay, more pathogenic than the gram positive. So if you take Escherichia coli, it will cause us the traveler's diarrhea. That means why it's called traveler's diarrhea means when traveler is going through several different places, the E. coli in the water, if it enters his body, it will cause us diarrhea. Okay, that's why it's called traveler's diarrhea. Gram negative. So several streptococcus and other varieties also there. So most of these gram negative bacteria are pathogenic. Okay, gram positive pathogenic also there, but comparatively rare, he told. Okay, don't gram positive should retain purple color? Yes. So this is the purple in their manner, and they actually change the color and all the things. So don't concentrate on this. Okay, gram positive should have the purple color. They should have colored some other things. Okay, they actually given blue and purple. It is so confusing. Okay, it's so confusing. Don't stick to this one. Okay, don't stick to these colors. What happens normally, it will be somewhat like this. It looks like this. Okay, gram positive will be look like this, and it may be this also, coccus or bacillus, it may be. And gram negative, we will normally use the saffron stain. That's why gram negative will looks like something in a red color. Okay, because we use a counter stain. Counter stain means another stain to notify that. That's we will use the saffron. Saffron will be red in color. So it looks, gram negative looks red and gram positive looks violet in color. Okay, which is having more lipid. I wrote down below. Gram negative will be having more lipid. So when you wash it with alcohol, alcohol will dissolve the lipids and the stains will wash away. Okay, so saffron, exactly, Lipika. Okay, so that's what happened. So one question came. So try to answer the question. Okay, simple. In fluid mosaic model of plasma membrane, upper layer is non-polar and hydrophilic. I don't know how many of you know this answer. And the polar layer is hydrophobic. Phospholipids form a bimolecular layer in middle part. Proteins form a middle layer. If you don't know, you can click on no option. Okay, in the participant list, and even in the YouTube also you can answer that. Okay, so some of you don't know, some of you know. Okay, then I will launch the poll. Shall I launch the poll or not? A fluid mosaic model. Why I am asking this question, because it's already, I launched the poll, try to answer. Okay, why I included this question, because this question is asked in CBSE 2002 paper. Okay, and I know that in CBSE this chapter is over for most of the schools. Okay, not all the schools, most of the schools. So Pawan, okay, so I think you guys can answer this, and I think, okay. Anyway, we are discussing about these things, yeah, but not in the prokaryotes. We will discuss about the fluid mosaic model and everything in eukaryotes, because plasma membrane shapes this is comparatively similar in prokaryote and eukaryotes for your level. Okay, for your level, it's same only. Really, Sachin. Great, bro. Okay, we could have also went in that way only. It will be awesome. Okay, I'm just waiting. I just want to see poll. Okay, so many of you are giving correct. Haha. Okay, some of you are guessing, but still correct. Okay, I'm ending the poll, guys. I'm not pressuring anyone. Okay, I'm just we'll see that. 20% choose an option A, 20% B, 45% C, and 14% D. Okay, so that's why like some of the things are become very important. Okay, so first tell me one thing. What is that thing? What is the meaning of polar? What is the meaning of polar? Is it hydrophilic or hydrophobic? Polar means hydrophilic or hydrophobic. What do you guys think? I think this is easy question. Exactly. Exactly. Everyone giving correct answer, right? Polar means hydrophilic, and opposite. You guys can think about it. Non-polar means hydrophobic. Okay. So now we will see. The fluid mosaic model, how it will be done? The fluid mosaic model is present somewhat like this. Okay. So think the plasma membrane is something like this. Okay. So there will be like two layers will be there, two layer of the lipids, and in that it will be called head. Okay, and it will be called a tail. Same thing with the another. This is called head, and this is called a tail. Okay, the head will be normally for polar. Okay, head will be polar, and the tails will be non-polar. Okay, head will be polar and tails will be non-polar. We'll see that now. Okay, upper layer is non-polar and hydrophilic, they are saying, but upper layer, as you can see here, it's the polar, right? You can remove this one. Polar layer is hydrophobic, they are saying. Okay, you can remove this one easily, right? Polar layer is hydrophilic, and phospholipids form a bimolecular layer in the middle part. They are saying phospholipids is form a bimolecular layer, like two layers. Okay, they are saying two layers it will form. So you may get confused, or some of you already got it. Okay, exactly. This one looks correct. And proteins form a middle layer. I didn't mention any protein here, right? I only mentioned the lipid only. Proteins will be present. Proteins will be present like this or like this or like this, but they never form a layer. Layer means it should be a sandwich, right? So the correct answer is option C. Okay. So this is what I wanted to discuss with you guys. Okay. So 1 second, guys. Yeah. So I'm launching NPS. You can answer the NPS, and then you just watch what is the homework for tomorrow. I'm damn serious tomorrow. Okay. Middle part means if it is something like this, if it is present something like this, then we call it as a middle part. Then, but there is no protein layer in the middle part, so no need to take tension. Okay. Homework, and I'm very serious. Okay. So what is your homework then? The first one, read Kingdom Monera. Our notes, okay. Our notes are there, right? What I wrote and what is the materials and all the things. Okay. And another thing, what you have to read, if it is completed, this chapter is completed in the textbook. So if chapter over, chapter cell chapter is over, read in CBSE or NCERT books. NCERT book. Okay. The very important another one thing. I don't know how many of you completed or not. Second chapter module. You have to complete it before you coming here. Okay. Complete it. I will include some of the questions from the module only. Then I will tell you. Okay. No, there is no biology test. Okay, polar repel, no, sir. Yeah. So we'll see that in when we are discussing about the plasma membrane, how that will happen. What will happen with polar to polar and non-polar to non-polar. Polar means which actually attracts or to the which actually loves to water, right? It will bind to the water like that. It's not like kind of liquid or anything. Yeah, I will tell you 1 minute. Sachin, next is the chemistry class, right? Ask that to Shiva sir. He will explain more properly than me. Okay, how the polar and non-polar word came from. I will tell you about the mentorship. I told you, Priyanka. I told you, right? Malpighian rule. That's what helped him to discover like acellula cellulae. That word. You can take screenshot only this much only. One minute. I will tell you about the JPMC and I will tell you about the mentorship program for like in 1 minute. So, any more questions about these things in this one? YouTube. Any questions, guys, in YouTube? So then Graham, who is he, sir? Graham is the scientist who actually developed the staining method. Okay, just remember about this homework. I will come back to that. Okay, 1 second, guys. Okay. Yes. Okay then. Okay, guys. If you don't have any doubts, YouTube, I will close the YouTube live for now. You can still ask the